Hello, BISC 210 students. Today we'll be going over Chapter 5 Genetics. In this chapter, we'll focus in on what is genetics. We'll talk about DNA replication, gene expression, meaning transcription translation. We'll talk about operons and how we regulate, or how bacteria regulate gene expression. And we'll talk about mutations as well. In this chapter, I don't cover every single thing. You're responsible for what I cover, not anything else in the book that I skip over. So if I mention it, it's fair game. If I don't, then you don't have to worry about it in the book. Alright, so let's get started. Genotype determines phenotype. So you may have heard these terms before, genotype and phenotype. But before we even get into that, let's start a little bit further back with a more basic definition of what genetics is and what genomes are. Genetics is the study of genes and their function and variations, what we call alleles, in genomes. Now, genes code for specific proteins, and then those proteins have certain jobs. When you refer to a genome, you're referring to all genetic material within a cell. And with bacteria, what's interesting about that is not only does it refer to the single circular chromosome, it also refers to some extracellular plasmids, which we'll talk about as well, which are pieces of genetic material that are not found in the chromosome. So now we know a little bit of a background. We kind of define these things. What is genotype versus phenotype? Well, genotype is actually the genes themselves. You look at our genome, we have a gene for eye color. We have a gene for hair color, shape of your nose, whether your ears are attached or detached. So those are your genotype, the actual genes. The phenotype is the outward expression. So what I can see when I look at you. So for instance, if I look at you and I can see you have blue eyes, or brown hair, or detached earlobes. What's that? That falls under the category of phenotype because it's this outward expression of the genotype. So I could see what the genotype actually is within the phenotype. Now, we'll talk more about this as we go through the chapter, but keep in mind genotype versus phenotype. So cells have DNA genomes, most of them, not all of them. Well, to clarify, viruses are technically not living, but some viruses actually have RNA genomes instead. So most cells are going to have DNA genomes, while viruses can have either DNA or RNA. Depending upon what virus you're looking at, its genetic material could be one or the other. HIV, for instance, has RNA as its genetic material, and when HIV infects human cells, it inserts the RNA, and then it goes backwards in terms of it has RNA, so it goes backwards and makes DNA, and then it goes forward to make RNA again, and then functional proteins. It's very, very weird, and really unique in that it goes backwards to go forwards. Now, this is obviously an exception. This is not a common thing. Most viruses are going to have DNA genomes. Bacterial cells, prokaryotic cells are going to have DNA genomes. Eukaryotic cells, DNA genomes. So more DNA as opposed to RNA. Now, one thing I will clarify here is viruses are not considered living. There's a lot of people who make the argument they are. There's a lot of people who make the argument they aren't. In general, we accept the fact that they don't fit the 5 characteristics of life, and they are technically not living. All right. Excuse me. The number of genes. So when we think about genes and the amount, we usually, for the most part, can safely assume the more genes, the more complex an organism may be. So, more genes, more complex organism. And a really great example of this is E. coli. The strains of E. coli that are non-pathogenic, which are the ones that you're going to get in your lab kits, have roughly 4,400 genes that we know of, that we've sequenced. But a pathogenic strain, a disease-causing strain of E. coli, specifically O157H7, which is the most pathogenic strain of E. coli that we will talk about, and we will talk about that in detail later in the semester. It has roughly 5,500 genes, so it has about 1,100 more genes than the non-pathogenic. And we can assume, since they probably have the same 4,400 genes as the non-pathogenic, those 1,100 that it has on top of the original 4,400 are probably associated with pathogenicity in some form, meaning they're associated with the reason why the organism is disease-causing in some way. Maybe not all of them, but I'd say a good chunk. So you can see non-pathogenic versus pathogenic. Pathogenic ones have a bit more genes than the non-pathogenic. Yeast cells. So yeast are unicellular. They are eukaryotic, which means true nucleus. Hopefully you remember that. They have about 6,000 genes. And then if you were to look at humans, we have between 20, they give 24,000 here. In reality, we have a range. We give between 20 and 25,000. We're not 100% sure the exact number of genes, because it's a lot. 20 to 25,000 genes is a whole lot of genetic material. So we give a range. The book says 24,000, but I would accept any answer saying it's between 20 and 25,000 if I were to ask that. But that's for human cells, so I probably won't ask that. So I mentioned that in addition to the single circular chromosome, prokaryotic cells can also have extracellular DNA or extra DNA or extra genetic material called plasmids. These are pieces of DNA that are outside the chromosome, and usually they provide a few genes that are extremely favorable to the cell. Whether that's genes that allow it to become pathogenic, sure. Antibiotic resistant, sure. Have the ability to create certain proteins or certain toxins, absolutely. So plasmids can allow for traits that the cell didn't have prior to obtaining that plasmid. Now, it doesn't always have to be traits associated with diseases. Sometimes plasmids carry genes that allow for metabolism of new substances. Let's say, for some reason, a bacterial cell could not break down fructose. Couldn't break down fructose. But all of a sudden it gets a plasmid that has a gene on it that allows it to break down fructose. So that's obviously beneficial. Additionally, not all plasmids are helpful. Sometimes cells will get these plasmids and they're just like, well, they don't help me at all. Oh well. And they're just there. But sometimes they can confer some advantageous traits. So hopefully in general biology you learned a little bit about DNA, how it's made up of, excuse me, it's made up of nucleotides. Specifically in DNA, it's made up of 4 of them. And we talked about earlier in the semester about organic molecules and their monomers, right? So here, for nucleic acids, we have nucleotides. And this is what a nucleotide looks like, right here. You have a phosphate group, which is the backbone. You have a sugar in the center, and then you have a nitrogenous base. Now, phosphate and the sugar stay the same. These do not get messed with. The nitrogenous base, on the other hand, in DNA, can be one of 4 different nitrogenous bases. It could be adenine, guanine, cytosine, or thymine. Adenine, guanine, cytosine, thymine. Those are the 4 nitrogenous bases found in DNA. If we were to look at RNA, there would be a slight change in the nitrogenous bases. One would get replaced. There are some big differences between DNA and RNA. So when you're looking at a structure like this, you can tell this is DNA immediately. Without even knowing what the nitrogenous base is, you can look at this and say, okay, I know this is DNA automatically. Why? Because on the sugar, each point is a carbon. So carbon 1, carbon 2, carbon 3, carbon 4. Okay? There's carbons at each point, except for this top one right here, that's an oxygen. I know by looking at this sugar, at the 2' carbon, the second carbon, there's only a hydrogen. There's only a hydrogen. Which makes sense if you think about the name of DNA, deoxyribonucleic acid. Deoxy, D means without, oxygen. So you're referring to the 2' carbon missing an oxygen attached to it. Then, if you had RNA, it would actually have the oxygen at the 2' carbon. Right here, it would be an OH group, called hydroxyl group. So you'd have an OH if it were RNA. But since it's DNA, it's missing the oxygen on the 2' carbon. So immediately I could tell which genetic material it is just by looking at the sugar. You can also tell the difference by structure. DNA is double-stranded, while RNA is single-stranded. Talk a little bit more about that in a minute. And nitrogenous bases, one of them is replaced in RNA. So the 4 nitrogenous bases in DNA are adenine, guanine, thymine, or cytosine and thymine. AGCT. However, in RNA, thymine is replaced by uracil. So in RNA, thymine is replaced by uracil. So if you're looking at DNA, you should be able to find adenine, thymine, cytosine, guanine. If you're looking at RNA, adenine, uracil, cytosine, and guanine. And the way I just listed them is how they pair up. So adenine in DNA, adenine and thymine pair up, and cytosine and guanine pair together. Now, hopefully from earlier chapters when we talked about proteins, you remember what type of bond holds DNA together. Hopefully you remember the two strands of DNA. When you have a cytosine on the left-hand side, on the left strand, and on the right strand you have the correct matching one, which is guanine, hopefully you remember that what holds them together is hydrogen bonds. So hydrogen bonds will hold the two strands together. Cytosine and guanine will be paired together with 3 hydrogen bonds. Adenine and thymine, 2 hydrogen bonds. Cytosine, guanine, 3 hydrogen bonds. Adenine, thymine, 2 hydrogen bonds. And you can see here, this is the structure, the double helix structure of DNA. And you have, this is the backbone, the purple piece. And then the lines are your base pairs matching. Okay. And then, remember, hydrogen bonds are holding these nucleotides together, the adenines and thymines and the cytosines and guanines. This is a close-up view of that. What I was mentioning here, you have your backbone, phosphates, your sugars, your nitrogenous base. Same thing here, sugar, phosphate, your phosphate backbone, your sugar, and then your nitrogenous base. And then the hydrogen bonds hold the base pairs together. So you have adenine and thymine with 2 hydrogen bonds, cytosine and guanine with 3. So if you were to try to break this strand, the more cytosine and guanine pairs that you have, the more difficult it becomes. So more cytosine and guanine pairs, harder it is to separate because of the fact that there's an extra hydrogen bond. Okay. One thing I will point out about this is these numbers that are here. On the left-hand side at the top you have this 5', at the bottom you have the 3'. Over here, it's the opposite, 3' to 5'. So DNA, the strands, are opposite of one another, meaning they have directionality. And that's important, because if the strands were to run in the same direction, it wouldn't be able to interact with the other strand. So imagine this. Imagine your 5' to 3', so these are your phosphate on the outside, your sugar, and then your bases facing inward. If this strand was running in the same direction, right, if we flipped it over, the problem would be the phosphate would be on the inside, the sugar would be here, and the nitrogenous base would be on the outside. So if we were to flip the strands, then that becomes a huge, huge, huge problem because of the fact that they won't be able to join together. But luckily, our cells flip one of the strands and have them running opposite directions, what we call anti-parallel. Parallel is when you have two lines running together in the same direction. When you have one going one way, one going the opposite, that's anti-parallel. And here's a little bit more about directionality. They use it with an example of a paper clip, which I really like. Shows you have a 5' end and a 3' end, and you're always going to have a 3 and a 5 interacting. So you have a 3' interacting with a 5'. And you can see here, by the time you get to step 4, you have a strand that goes from top to bottom, 5' to 3', and then you have an opposite strand that goes 3' to 5'. And what that does is it allows the phosphates to face outward on both strands. It allows the sugar to be within the backbone, and then it allows the nitrogenous bases to face each other. Because if they don't face each other, then they can't link together. So this is a really great image showing you directionality. Directionality is going to be very important when we talk about specific functions like transcription and translation and replication. All of those will be required to understand directionality. And again, this is just what I was mentioning with anti-parallel. Here are some key differences between RNA and DNA. I've went over most of them, not all of them, but the highlights, RNA, you have uracil instead of thymine. So adenine in DNA pairs with thymine, but in RNA, adenine pairs with uracil because uracil replaces thymine. There is directionality. Typically, you're going to find it in a 5' to 3' direction, RNA at least. DNA, you'll have two strands, one 5 to 3 and the other 3 to 5. RNA is not always single-stranded, but quite often single-stranded. There are examples of double-stranded RNA, but for the most part, we only focus on the fact that it is single-stranded. We really won't talk about double-stranded RNA in this course. The fact that it can fold on itself and form helical and loop structures, that's interesting, but not a big deal. We're not going to worry about that. But the ones above it, the three above it, 1, 2, 3, all three of those are worth noting. There are three major types of RNA that a cell has that we will talk about. We only talk about two of them. The third one is not totally necessary for us to focus on in this class. So messenger RNA is this molecule that's created during transcription. So you take your DNA, it's read, and then we make a shorthand copy of the gene of interest. That shorthand copy is called messenger RNA. Then, when we move into translation, transfer RNA helps build a polypeptide, a protein. So these two RNA types are very important to gene expression, transcription and translation. Now with transcription and translation, the way I'm going to do that in this class is I'm going to go through this entire PowerPoint, cover transcription translation pretty generalized, And then I'm going to use a whiteboard function, and I'm going to draw it out step by step. So we won't necessarily go through every single detail this first time through, but I will create a complementary video of both DNA replication, DNA transcription, excuse me, and translation. So I will go through all of that. You'll have all of that detailed to you. The central dogma of biology. Hopefully you know this from general biology, whatever you took it last, is DNA, RNA, proteins. You start with DNA, you end up with mRNA, and then you end up with a functional protein. And here, what's interesting, it shows you you go from DNA, you undergo transcription to make your mRNA copy of your gene or genes of interest, and then you translate that by creating a functional protein. Notice it mentions reverse transcription. This is talking about one of the prime examples is HIV. HIV has an RNA core. It has RNA genetic material. So it infects the cell with RNA, but it goes backwards and makes DNA, and then goes forward, going through transcription to make mRNA, and then translation through to make a functional protein. The mechanism behind the fact they go backwards to go forward is really unique, but it's a little bit outside of what we are going to focus on in this class. We'll mention HIV, but we're going to talk more about other pathogens. Now, DNA replication. So when cells need to copy their genetic material, they go through the process of DNA replication. Basically, right before in bacteria binary fission, cells have to copy their genetic material, right? Because they're making two new daughter cells that should be genetically identical, so they go through DNA replication to make identical genetic material, so the daughter cells can be genetically identical. In bacteria, it's right before binary fission. In humans, it's right before, hopefully you know what process. What process do our cells use to replicate themselves? Hopefully you answered mitosis. In humans, it's mitosis. But in bacteria, it's binary fission, and they basically, it's a more simplistic version of mitosis. So they, in order to replicate the genome, to copy it exactly, there are some really important functions that need to happen. Otherwise, it is not going to work. First, you have to unwind the original DNA. Remember, DNA in bacteria is extremely coiled, right? We talked about how a rubber band, it's one giant circle, but it twists and turns and folds in order to take up less space in the nucleoid region within the cell. So once you're done unwinding the original DNA, then you got to copy it. You got to go through replication. And then once you're done, you have to rewind or put the DNA back together, and then you have a newly made strand or newly made copy as well. So then each new daughter cell has the same genetic material as the other. This is what DNA replication looks like. You have two what we call replication forks. Remember, bacterial genomes are circular, so this is accurate. And you have two replication forks. meaning you have two positions on the chromosome where replication is taking place. So replication is taking place in this spot right here, going this way, going up. And then there's one down at the bottom of this bubble, and it's going down. And what they do is they continue to go around and around and around and around, and eventually the fork on the top meets the replication fork at the bottom on the other side of the strand. So think of it this way. You're on a track, and you and your best friend are standing at the exact same spot. One of you starts running to the left, one of you starts running to the right. And if you're running at roughly the same pace, you're going to meet on the other side of the track, if it's an oval-shaped track, on the other side of the track about halfway, right? Almost to the exact same position of where you started, just on the opposite side of the track. That's what's happening here. This is going up, and it'll come around, and it'll meet this other strand that's going down right around here in the middle. Once that happens, the two pieces of DNA, the new strands, can separate, and you have one copy that'll go to daughter cell 1, and you'll have a second copy that'll go to daughter cell 2, and they are both identical, barring any random mutations, spontaneous/random mutations. If there are none of those, then they are genetically identical. Now, the color of the strand actually matters, because what they're saying with that is DNA is a semi-conservative process, and we'll talk more about that in a minute. Here's a closer look at a replication fork. This is one replication fork. Obviously, if this is the one facing up, okay, that's fine, but there's also one facing downward. Now you can see there are two strands. It opens the DNA, and there's a strand here, there's a strand here, and there's a lot of stuff going on. There's a lot of enzymes that are getting involved in this process. Okay, there's a lot of enzymes that are getting involved in this process. You will end up having to know these enzymes, but at the moment, we're not going to really go into all of them. We're just going to. I'm just showing you the fact that you have both sides of the DNA, both strands are being utilized. You have this strand being utilized and this strand being utilized. Because the whole point of this is to make, at the very end, two identical daughter strands. Right? So you'd have an identical copy that you've made, and hopefully they're genetically identical and there's no spontaneous mutations, which are pretty rare. When going through replication, mutations can occur, random mutations, or mistakes. So, for instance, there's an enzyme called polymerase that uses the template strand, which is the pink one, to build a new strand that matches it. Sometimes polymerase messes up. Sometimes instead of putting, let's say there's a G and a C on this pink strand, instead of giving it a C and a G in the correct order, let's say it gives it an adenine and a thymine. That's wrong, because we know that adenine and thymine pair together, but they do not pair with cytosine and guanine. So accidents like this, spontaneous or random mutations, can occur, but they are extremely rare, and when they do happen, they can be fixed. They can be fixed. And later in this chapter, we'll talk about how they actually fix any mistakes that are made. So this is in your book, this image. I don't have the image number here, sorry. Oh, yes, I do. 5.8. Take a look at it. I will be drawing it later on a whiteboard function, and we'll talk about each enzyme that's associated with it. Now, this is a very important concept. We need to make sure we understand why replication happens. DNA replication happens because the cell is about to undergo binary fission, and it needs to make two identical copies of the DNA for the two new daughter cells. Okay, so DNA replication is copying the DNA so the two new daughter cells have the exact same genetic material. Why am I going over this and repeating myself over and over again? Because we're going to talk about some more processes that occur that don't have to do with DNA replication, but they are still very, very important. Replication is a semi-conservative process, and by the end, there are two separate chromosomes. In bacteria, two separate circular chromosomes. Now, I will explain semi-conservative a little bit in more detail later on when I draw out the process of DNA replication for you. But it is a semi-conservative. And what that really means is it uses some of the old DNA to make some of the new DNA. So it uses the old strand to help make the new strand. Here, as I mentioned, replication, there can be mistakes. Random or spontaneous mutations can occur. However, that's pretty rare. And if you look at this example, E. coli can copy 500 nucleotides per second. That's a lot. And it makes an error about once every 10 to 100 billion base pairs. So 10 to 100 billion nucleotides. That's rare. It is very rare. But the cool thing is if they screw up, there's a mechanism that will allow the cell to fix that problem. It will cut out the wrong nucleotides and then replace them with the correct ones. Okay, so this is the end of me talking about DNA replication for now until I start doing my drawing for you. We're going to move into, at this point in this chapter, gene expression. So protein synthesis and gene expression. So we know that we start out with DNA, and the DNA is kind of like a cookbook. It has all the recipes to every protein that needs to be made. It has all the recipes that the organism needs to make proteins, functional proteins. So, with protein expression, basically, you take the DNA, they choose whatever gene they want to express, they make an mRNA copy of it using transcription, the process of transcription. And then the mRNA copy is kind of a shorthand version of it. You add the amino acids in as your ingredients, so to speak, and then you use translation in a ribosome to build a functional polypeptide or a functional protein. That sounds all well and good and easy, but there's a lot to it. And again, I'm going to kind of skim over it in this video, but I'm going to provide you a second video where I go into great detail about replication, transcription, translation. So there are several components that a cell needs to undergo transcription. These are some of the enzymes. So if something ends in ACE, A-S-E, it is almost always, not always, but almost always, an enzyme. It ends in ACE, it's almost always an enzyme. RNA polymerase, gyrase, helicase. A promoter is not an enzyme, it's a region on the genetic material. So this is kind of transcription in a nutshell. I know it seems really confusing with all the information here, but honestly, it's pretty easy after you've gone over it a couple times. So we'll focus right here. Here's where the gene of interest is. Let's say this is the gene that allows the cell to make lots of sucrose, and the cell needs lots of sucrose. So at this point, it will transcribe, it will transcribe this region, making an mRNA copy, a shorthand version of the gene of interest, and then that mRNA, which right here is, here's your final product, your mRNA strand will then go through a process called translation, where a ribosome will read the mRNA strand and build a polypeptide. Now, there are quite a few bits of information in here that I'm going to have to focus on when I start drawing these images out as opposed to right now. But this gives you a general idea of how this is happening. You find a gene of interest, or the cell has a gene of interest. It starts to use a polymerase to read the strands for that gene of interest. It'll make an mRNA copy. Once that mRNA copy is made, then they move into translation, and translation will make a functional protein from the mRNA. Sounds all well and good. Can get a little confusing. I do want to point out that in bacteria, transcription is occurring, translation is happening at the same time. So you can see this RNA right here, this mRNA is being built. So it's not fully built yet because this is the product. But notice it's about halfway, I'd say. As this thing is being built, it's also being read and translated at the same time. Why? It saves energy. If you do things in sequential order sometimes, it takes too long. So if you undergo transcription and then you wait and wait and wait and say, all right, it's finished, now I can undergo translation, sometimes that's too long. So in many cases, transcription and translation happen simultaneously. As the mRNA is being built, it is also being translated into a functional protein. I will focus more on that a bit later on. So, genetic codes and amino acids, there are 4 nucleotides in RNA. There's also 4 in DNA. But the 4th one differs depending upon whether you're looking at DNA or RNA. If you're looking at RNA, it's adenine, uracil, guanine, and cytosine. So uracil replaces thymine. So that means it pairs with adenine, and it pairs with adenine with 2 hydrogen bonds. Where the other 3 pair together, and they each have 3 hydrogen bonds. So, when you have mRNA and the ribosome is reading it to build a polypeptide, it reads it in codons. And what a codon is a triplet of nucleotides, meaning it's 3 nucleotides. So it looks at a codon, a triplet of nucleotides, and that triplet codes for a very, very specific protein. Excuse me, I'm sorry. Let me correct that. Very, very specific amino acid, which is the monomer of a protein. I apologize. So it codes for a very, very, very specific amino acid. Now, there are 64 codons, 20 of them, or 22 of them, we have 22 amino acids. There are 3 nonsense codons, what are we call stop codons, and 1 start codon. So there's 60 that are actually going to code for things. They're going to code for amino acids. There's 3 of them that act as stop signals, and then there's 1 of them that acts as a start signal. Now, there's a lot of redundancy when it comes to transcription, or excuse me, translation and reading the mRNA code. So what I mean by that is, if you look at this table, which is in your book, notice that serine is coded for 2, 3, 4 times, 5, 6 times. There's 6 different ways to code for serine. For leucine, there's 1, 2, 3, 4, 5, 6. So there's 6 ways to code for leucine. For others, lysine, arginine, there are multiple ways to code for a single amino acid. So there's a lot of redundancy. So if you were to look at the sequence on the mRNA strand and see CUC, you get leucine as the correct amino acid that goes along with that. But in the same breath, if you got UUA, you would still also get leucine. Which is why, in some cases, when mutations happen, they actually go unnoticed in some cases. Because let's say you had, CUG. Let's say this was the codon, and then a mutation occurred, and it changed the C to a U. So now you had UUG. Well, guess what? You still code for leucine. It doesn't matter whether it's CUU or CUC or CUA, CUG, or UUA or UUG. Who cares? It really doesn't matter because they all code for the same amino acid. So if there's a change and it still codes for the same amino acid, we call that a silent mutation because it doesn't make a difference. Yeah, there's a change in the sequence, or there's a change in the original sequence. However, it doesn't matter because it codes for the same amino acid. Now, I don't expect you to memorize all of these, but there are 4 that I do expect you to memorize, and those are the stop codons and the start codon. So the start codon is always AUG, methionine. AUG, methionine. The stop codons are always UAA, UAG, and UGA. So UAA, UAG, and UGA. They do not code for anything. They just signal that that's the end of the gene that needs to be translated. So once the ribosome reaches a stop codon, it stops translation. It says, all right, I'm done. I've translated the whole thing. And it stops. Now, again, they don't code for any particular amino acid. They just code for a stop for the ribosome. It hits one of those 3 and it says, all right, I'm done. I've completed it. I've translated the whole thing. So, just to clarify, 3 nucleotides equals 1 codon. So 3 nucleotides is the equivalent of 1 codon. 1 codon codes for 1 amino acid. And that's the way you should look at it. When you're looking at translation and you're looking at the sequence, you will read 3 of them at a time. 3 nucleotides equals 1 codon. We read 1 codon and it codes for a single amino acid. This is what I was talking about, redundancy. Just 1 amino acid can be coded for multiple ways. And I have already mentioned leucine, where it can be coded for 6 different ways. This is just talking about how certain organisms can build proteins really fast, and it rarely makes an error. 1 in about 10,000 codons, that's pretty good. It's actually really good. And it can be fixed. So it's a pretty good setup. Humans are very similar as well. We have redundancy. We also have the ability to fix errors as they pop up. So, translation. This image here is actually showing you both transcription and translation. Excuse me. You would have here, so it's showing you transcription in that here's your DNA, right? Here's your DNA, which is going to get read by a polymerase, and as it reads, it builds an mRNA strand, right? It builds your mRNA. As your mRNA is being built, multiple ribosomes attach and translate this mRNA as it's being built. And it does it in chunks. It does it in chunks. And then the growing proteins, or the polypeptides that are built, will eventually be brought together and create one functional protein. So why in the world would it be efficient to undergo transcription and immediately at the same, almost at the exact same time, start translation. It all comes back to energy and energy conservation. All comes back to energy and energy conservation. If you can transcribe and then translate really fast, saves a bunch of energy in the long run. Also, some of those proteins may be needed immediately. So, as the mRNA is being transcribed, being created, it is also being translated by ribosomes, multiple ribosomes. This image shows, on the right here, the pink is the DNA. There's a yellow line in the middle here. That's the mRNA strand that's built, and here's another one that's been built. And all these greenish teal blobs are ribosomes. So as the mRNA is being built, all these ribosomes attach and build a section of that mRNA strand. They build a section of the polypeptide, and once they're all done, they piece the polypeptide together to make one functional protein. So it's happening simultaneously. Energy conservation. So this is another look, a closer look at translation. This is how they actually build the building the amino acid sequence. You have a ribosome, a molecule called tRNA comes along, and it brings the correct amino acid to match the codon on the mRNA sequence. So let's say this one right here was on the mRNA AUG. The tRNA has the anticodon, meaning the one that matches it, with the correct amino acid. So this tRNA would have, if this sequence was AUG, what would be the matching sequence to that? What pairs of adenine in RNA? Be uracil. If you have uracil, what would you have? What was the opposite of that? Adenine. And then if you have G on the mRNA, your tRNA is going to have C. So it'll be UAC is the match, is what we call the complementary codon. And it brings the correct amino acid based on that chart that we looked at. Well, UAC codes for the first amino acid, which is methionine. You'll always see AUG, that's where it starts. So you'll see AUG on the mRNA, and then the tRNA brings UAC, the matching complementary codon, and the correct amino acid, which in this case is methionine. Then the ribosome reads the next three. And let's say it says GGG. The tRNA that has the anticodon, or the one that matches it, CCC, will bind and it'll bring the correct amino acid, whatever that amino acid is. We don't need to memorize all of them, right? I told you that when we saw the chart. But it brings the correct amino acid, whatever that amino acid actually is. And this is how a polypeptide starts to get built. The ribosome keeps reading and it keeps adding amino acid after amino acid after amino acid until eventually it hits a stop codon. UAA, UAG, UGA. One of those three. Here it hits a stop codon, UGA, so right here, UGA, there's a termination factor there. And when the ribosome hits that termination factor, the polypeptide is released. The ribosome separates, it lets go of the mRNA strand. And then, on top of that, the tRNAs float away and get utilized again elsewhere. And what you have left is your primary structure, your polypeptide, right? The primary structure of a protein. All it is a series of amino acids in a chain. That's your primary structure. At this point, proteins are not functional. Proteins are not functional. Proteins have to fold into a very specific shape in order to be functional. Proteins have to fold into a very specific shape to be functional. If they do not fold into that shape, then they can't be used. So, this released protein will have to fold into the correct shape in order to become active and functional. Again, I am going to draw transcription, translation, replication, as well as a couple other processes after I've gone through this chapter. So what about protein synthesis do we need to think about in terms of rate of reaction? We know proteins are essential. We know we make them through transcription and translation. However, making those proteins is extremely expensive in terms of energy. Extremely expensive. So the cells want to be A, as efficient as possible, and B, be able to regulate when they take place. A good way to think about this is if you have too much of something, right, you don't need more of it. What you want to do is you want to use what you have in front of you, and then once that stockpile gets too low, then you get more of it, right? So let's say I have a bag of peanuts, okay? I put them on the table, and there's 100 peanuts. These numbers, by the way, are arbitrary. They don't really mean anything. But let's say, all right, I got 100 peanuts. That's more than enough peanuts. I don't need any more than that. So instead of asking for more or getting more, I don't ask for more and I don't get more because there's no need to. I have enough, right? So then I start to eat the peanuts. Okay, I'm eating them, and eventually I eat 90 of them. Okay. Yes, that's a lot of peanuts. But let's say I eat 90 peanuts, and all I have is 10 left. And I'm still hungry. So what I can do at that point then is then I can ask for more. I can say, okay, I need more peanuts now, and then replenish what I lost or what I consumed. Same kind of concept here. When it comes to protein synthesis, they will make the protein as often as possible until they have too much. Once they have too much, cells have to figure out a way to say, all right, please, let's stop making more of this product because we just have way too much and it's wasting energy. Energy is a valuable commodity when it comes to cells. So saving it is extremely important. So when they have too much of something, they say, all right, I have too much of this protein. I need to slow down making more of it, or I need to stop making more of it completely. So one or the other, there's a way to control gene expression and its rate. In addition to being able to control specific genes and say, okay, well, I've already made too much of gene A, let's slow it down for now and then we'll pick it up later on. Well, there are other genes that don't even get activated at certain points. They leave them inactive. So what's the point in making a gene that has something to do with binary fission when you aren't going, or when the bacterial cell is not going to go through binary fission for another 8 days, right? Timing is kind of arbitrary as well. Some bacteria undergo binary fission every 20 minutes, 20 to 25 minutes. But let's say this one undergoes binary fission every 8 days. The genes that need to be turned on for binary fission to happen, well, the 7 days before binary fission occurs, they're turned off. They're not functioning because it saves energy. There's no reason to have those genes running and making proteins when you're not going to go through binary fission for days, okay, or extended periods of time. When I say you or you are not, I'm referring in terms of bacteria. So anytime you hear me say you, I'm really saying bacteria. Now, one way to control gene expression is through operons. Bacteria, this is unique with bacteria. Operons are essentially regions on the chromosome where genes that have similar functions get placed together. So, for instance, on a chromosome, in one spot, you may have 8 genes in a row that are all associated with the formation of the cell membrane, which means they would all get transcribed and translated together. So in operons, the goal is to group together genes that have very similar functions so they can be transcribed and translated at the same time. And a really good example of that is traits that make cell walls or cell membranes. They're all grouped together, most of them, sometimes not all, but most. And you have, let's say, 8 or 9 of them all grouped together. They all get transcribed together, they all get translated together, which makes it more efficient. Whereas in humans, the gene for eye color is affected by multiple genes. It's not just one gene, it's multiple genes. And they're all on, most of them are on different chromosomes. And if you recall, we as in humans have 46 chromosomes or 23 pairs. So eye color, that's a bit more complex because it's multiple genes that control eye color, and they're all potentially on different chromosomes. But operons for bacteria, group them all together, which makes a lot of sense and makes it way more efficient. There are some really important parts to operons: the promoter, the actual genes themselves, a repressor, and an operator region. So though I will be drawing it out in a bit, we're going to kind of look at this as well. Excuse me. Before we do, I want to talk about two types. There is inducible and repressible. We're not going to talk about inducible operons in this class. There are a bunch. Inducible simply means they're off by default and have to be turned on. So in that operon, in an inducible operon, where there's however many genes that have similar functions are all grouped together, but in an inducible operon, transcription and therefore translation are turned off by default, and it has to be turned on. You have to induce transcription. Repressible operons are the opposite. The operon is always on, therefore transcription and translation are always happening. But when we don't want it to happen, we have to repress that operon. We have to stop transcription and therefore stopping translation. So we have to shut it off. Now again, this is all happening in bacterial cells. When I say we, I mean bacterial cells. Have to shut it off if it's a repressible operon. If it's always on, going to be spending a ton of energy. And if those proteins don't need to be made at that given moment, then you're wasting energy. So, in a repressible operon, it gets shut off. Or inducible gets turned on. Great example of a repressible operon is the arginine operon. Its job is to regulate the production of the amino acid arginine. Now, the arginine operon being repressible means it's on all the time until it is told to stop being transcribed and translated. So this goes back to kind of like my peanuts example. If the arginine operon is always on, it's making arginine nonstop. If you get too much arginine produced, then the cell can't use it all fast enough because it keeps replenishing all that arginine. So to slow it down, they repress that operon. Okay, it's repressible. They turn it off, which means no more arginine will get made. And then the cell can use the stockpile of arginine that it has until there's very little left, and then they can restart the arginine operon. They can restart the arginine operon. So they use their stores, however much they have stored, first. And then, after that, they then will turn the arginine operon back on after they've used up almost all of the arginine available to them. So that is a repressible operon. I will talk more about it when I start drawing stuff for you. So these are some of the steps. Arginine binds to the repressor. So when arginine is available, you have a lot of arginine. The product can actually activate what we call a repressor. So arginine can activate a repressor. Repressor is a protein typically. And the repressor protein, once the arginine binds to it, has the ability to bind to the operon, that region, the operator region, and it will stop transcription. So the product itself, arginine, is inhibiting more production of itself, which makes a lot of sense if you think about it. If you have too much of something, sure, it could be used to say, all right, well, I have too much of this. Let me help activate a repressor protein and stop making more of it because I already have too much. So, very important. Please, if you're confused on any of this, definitely come to office hours, go to SI. This is all really, really important, and you will be tested. This is an image in your book that I like a lot. So this shows you a repressible operon and the fact that it's always on until we turn it off. So here's the DNA. Here is the region where you have the genes that get transcribed. And here's the polymerase that will do the transcribing. Notice there's very little arginine, so it's okay to make more, to let this continue doing its job, because they don't have enough. There's very little arginine. So this polymerase continually, or a polymerase, will continually, over and over and over again, create more and more and more arginine. But eventually, when there's too much arginine, like over here on the right, the arginine will trigger a repressor protein to activate. The repressor protein will bind to a very specific region called the operator region, and it blocks the polymerase from transcribing those genes. So it's physically blocking the polymerase. Now, the cell has to use the stores of arginine until they get really low. When they get low again, this repressor right here that's stopping the polymerase will pop off, it will let go, and it will allow for transcription and translation of arginine. So it's not a always turn it off, it's off forever, or turn it on, it's on forever. Nope, this can happen. We can turn it on and off, on and off, on and off, depending upon, in this case, arginine concentration. Now this is just one example of a operon. There are tons of others. There are other amino acid operons, like the lac operon. There's also operons associated with cell membranes, some associated with pathogenic traits. So it's not just arginine, but arginine is a really good example of a repressible operon. And again, I will be drawing this in a little bit. So, issues with translation. So mutations are simply defined as the change in genetic material of a cell or a virus. Any change. Okay, there are different types, but any change in the original sequence is considered a mutation. Now, substitution mutations are when you accidentally put an incorrect nucleotide down. So let's say the polymerase is reading, it sees a adenine, it sees a cytosine, and it's supposed to put a guanine to match it, but instead it puts another, let's say it puts an adenine instead of a guanine. That is a mistake. That's a substitution mistake. Insertion is when you add extra nucleotides to the sequence. So somewhere in the sequence, one or more nucleotides will be added. Deletion is where somewhere in the sequence, one or more nucleotides get deleted. When it comes to insertion and deletion mutations, they lead to something called a frame shift mutation. So if you have an insertion or a deletion, it leads to what we call a frame shift mutation. If you have a substitution mutation, and the substitution mutation, if the change leads to the same amino acid being coded for, it's considered silent. Because remember, there's a lot of redundancy, right? Serine can be coded for 6 ways. Leucine can be coded for 6 different ways. So in a substitution mutation, if the nucleotide is changed and it still codes for the same amino acid, it's considered silent. Then there are 2 others. If you have a substitution mutation and it changes the nucleotide, and now all of a sudden you have a new amino acid that's coded for, that is called a missense mutation. Missense mutation. So for instance, let's say the sequence is CCC, and the middle one changes, and now it's CAC. And all of a sudden, the original amino acid coded for was CCC, was serine, but now that it's CAC, it codes for isoleucine. It codes for a different amino acid. That is called a missense mutation. Missense. If there is a substitution mutation and it codes for a premature stop codon, UAA, UAG, UGA, it is called a nonsense mutation, because the process of reading that sequence is going to stop in a random place. It won't be completely read, which means the protein will be useless. So that is a nonsense mutation, which comes from a substitution mutation. So here are a couple examples. This is the original, so original DNA, TTT, no changes. This is the codon in mRNA, AAA, and it codes for lysine. Perfect. Then, notice change from TTT to TTC, and then the codon changes as well to AAG, but it still codes for lysine, which means it's a silent mutation. If you have TTT, all of a sudden that middle one changes to G. Your mRNA sequence changes from AGC to ACG or ACA, excuse me, ACA, and it codes for a new amino acid that is called a missense mutation. And then lastly, if there is a substitution mutation that codes for a premature stop codon, it is called a nonsense mutation. Premature stop codon, nonsense mutation, and it has little to no functionality. Little to no functionality. Okay. This one, I want to talk about another type of mutation that can occur, and it's actually very detrimental. This is the normal sequence here on the left. There's an insertion, meaning there's an extra nucleotide that's been added. And the way we read the sequence changes. So normally, the original sequence is TAC-TTT GAA-TAT-ACT. That's how we would read it 3 at a time. TAC, TTT, GAA, TAT, ACT. But if we throw a extra nucleotide somewhere in there, everything after the inserted nucleotide will shift to the right. However many spots. If you insert one nucleotide, they'll all shift over to the right one spot. If they insert 5 nucleotides, then everything will shift over to the right 5 spots. But now the problem becomes we read the sequences very differently. We read the beginning, TAC is the same, but now everything else after that will potentially be different. So now it's ATT, TGA, ATA, TACT. So you can see how inserting a nucleotide shifts everything to the right that many spots. If you insert one, it only moves over one spot. But it drastically affects the way you read the sequence. And in this case, it actually leads to a premature stop codon. Therefore, the protein doesn't get made. In this case on the right, it's deleting a single nucleotide. So what will happen is everything to the right of that deletion will shift to the left that number of spots. So we deleted this T, everything to the right of it shifts to the left one spot. So now when we read it, it is very different than the original sequence. And you can see, look at the amino acids that are coded for. They are very different. Yeah, methionine stays the same, but then you have asparagine and then leucine and then tyrosine, whereas you would have lysine, leucine, isoleucine. So pretty different. Pretty different. Anytime you have a addition or deletion mutation, it is considered a frame shift. It shifts the way you read the sequence. We call it a frame shift mutation. It affects the way you read the sequence. The amino acid sequence is different. Okay. Now, we've gone through all the examples of mutations, whether that's a substitution, or an insertion or a deletion, we've seen that they can have different outcomes. Mutations can be helpful sometimes, they can be detrimental, or they can be neutral. So they vary in their effect. Most are neutral. But we went through all of these. Silent mutation, there's a change in the sequence, but the amino acid stays the same. Nonsense, there's a substitution, but it codes for a premature stop codon. Missense, there's a substitution, and it codes for a new amino acid than the original, compared to the original sequence. Frame shift, you either insert or delete nucleotides, one or more, and the sequence shifts, and the way you read it becomes drastically different. And with frame shift, there's almost no way to save a protein after a frame shift mutation. Because the way you read it is so different, it's just not going to be the same at all. And therefore it's useless. I've talked about spontaneous mutations. These are just random ones that happen but are very rare. So, spontaneous mutations, they happen, it's rare, okay? There are mechanisms to prevent this from happening. Now, spontaneous just means it happens because it happens. But mutations can also be induced, meaning something can trigger them. And one of the ways they can get triggered is by a mutagen, which is either a chemical, physical, or biological agent that increases the rate of mutation. So, for instance, UV light, so sunlight, it can lead to changes in the changes in the sequence, which the longer you're out in the sun, the higher the risk, because it is considered a mutagen. With UV light, it creates something called a thymodimer, which I'll talk about later, that cross-links the DNA and messes up its ability to undergo transcription. Okay. This is why I said spontaneous mutations, random mutations are pretty rare, because polymerases, which do the reading during transcription or during replication, they have proofreading mechanisms, meaning they say, all right, I'm supposed to put TTG, and instead they accidentally put TGT. They can look back at that and say, whoops, I made a mistake. And it will cut out the mistake, and it'll replace it with the correct nucleotide. So it'll cut out the mistake and fix it. Polymerase, 1 in every 10 billion nucleotides or base pairs, there's an error. That is extremely rare. For humans, 10 billion's not a lot. We have a very large genome. But for bacteria, 10 billion base pairs, that's pretty high. Especially when you consider some pathogens or some bacteria even just have 500, 700 genes total. They have 500 or 700 genes total, It may not even have 10 billion base pairs. I mean, obviously this is a range. It's an average, but on average every 10 billion base pairs. But I mean, when you don't have 10 billion, hopefully the proofreading mechanism works well and is functional. You can fix some mistakes. This is one of them I was talking about. Excision repair, basically, they put down the wrong nucleotides, they chop them out, and replace them. So, put down the wrong nucleotides, whoops, it happens, but so it happens, but it can be fixed. They can cut them out, and then they can, after they cut them out, they can replace them with the correct nucleotides. Here's an image of showing excision repair. You have what we call a thymer dimer. If you have several thymines in a row, they cross-link, meaning they'll attach to the one next to it, creating a little bit of a bubble. And when polymerase goes to read this, it actually can't go over the bubble. It gets stuck in front of it. So the excision complex will chop that region out, and then another polymerase will come in and fix it, put in the correct nucleotides, and then an enzyme called ligase will tie it all back together, and you get one continuous strand. One continuous strand. Okay. So we've gone through mutations, we've gone through transcription, translation, we've mentioned it. Now I want to talk a little bit about how bacteria get genetic material that's extra. There's two major ways that you have gene transfer. You can have vertical gene transfer or horizontal gene transfer. Vertical gene transfer is simply undergoing binary fission and creating two new identical daughter cells. So the information for the daughter cells are the same. It's passed through binary fission. Horizontal gene transfer is a bit different. Horizontal gene transfer allows genetic material to be passed between cells that's not associated with binary fission. So horizontal gene transfer allows for genetic information to be shared, not through binary fission. Vertical gene transfer is sending genetic material from the parent cell to the two daughter cells. But horizontal, it has nothing to do with binary fission, and it comes from other cells. Okay. So, three major types: conjugation, transformation, and transduction. These are the three major types of horizontal gene transfer. Conjugation, basically it has what we call the F plasmid, the fertility plasmid, and it creates a tube called a pilus that will attach to a neighboring cell. So it's pretty much like bacterial sex. The bridge goes from one cell to the other, and it will then transfer the F plasmid from one cell to the other. Once the F plasmid has been transferred, the pilus breaks apart and the cells go on their merry way. So conjugation is similar to what we would say bacterial sex, creating a pilus which attaches and they send the F plasmid to the other cell. So it can then transfer to other cells. That's what it looks like here. And it's going to transfer the F plasmid from one cell to the other. Here's another way to look at it. This is one of the ways your book has it laid out. Here's your F factor, fertility plasmid. This is a cell without that F factor. This one creates this pilus, which will then attach to the cell without the plasmid, and then it will make a copy of the F factor and send it to the cell that didn't have it before. Now they both have it, and they can then pass it along to other cells. Both of them can. Transformation. Transformation is when bacteria take DNA up from their environment. This is pretty neat. So let's say in a solution, and I've done this in a lab, you have bacteria in a solution, and what you do is you allow for the bacteria to take any DNA that's floating around in the solution and incorporate it into its own cell. So let's say there's an E. coli cell in some solution, and there's DNA floating around that has genes on it that E. coli doesn't have. Well, that cell can then pick up and absorb the DNA through the cell wall and incorporate it into its own genome. And now it has a new function. It has a new trait that it didn't have before. This is really neat. Only catch with this is in some cases, actually many cases, you have to manipulate the cell wall a little bit. There's several ways to do that. You can do it chemically, or you could do it one way we did it in our research lab was we used sonic vibrations. So basically, we would create this high frequency wavelengths, this loud noise, and it would kind of pop holes into the cell wall, and the DNA would be able to enter through those tiny little holes that we would generate. Very interesting. But then the cell would actually have new traits to incorporate that into its genome. There's a really important experiment that occurred that proved this, and it's called Griffith's experiment. Basically, these are the steps that happened. Now, I'm going to move to the next slide because there's an image, but you can come back to this slide and see exactly word for word what is going on during each step. So scenario 1, you have Strep pneumoniae, which is an encapsulated pathogen. Okay, it's alive. Okay, it's surrounded by a capsule. And when it enters the mouse, it killed the mouse. Then in scenario 2, living non-encapsulated strep pneumoniae, basically they got rid of the capsule, but the cells are still alive, strep pneumoniae. Enter the mouse, mouse is fine. Mouse is healthy. Third scenario, You take encapsulated strep pneumoniae and you heat it to kill it. Once it's dead, you then inject it into the mouse. It's dead, therefore the mouse is perfectly fine. But here's where it gets interesting. In this last scenario, if you inject live non-encapsulated strep pneumoniae and heat-killed encapsulated strep pneumoniae, okay, this is dead, but it has the genes for the capsule. So what will happen is the living non-encapsulated cells, through transformation, can pick up the genes from the dead cells to form capsules, and you can see that down here, and kill the mouse. So when you heat kill the encapsulated strep pneumoniae, the genes, the genetic material associated with the capsule being formed is free-floating, and this non-encapsulated version will pick it up through transformation, and now it has the genes to create the capsule, which it then does, and you can see from the image on the bottom right here, which will then kill the mouse. So even though the mouse was not exposed to a live encapsulated strain, which is the deadly strain, they still formed. And that is because the genes that were released from the heat-killed encapsulated strep pneumoniae were picked up by the living non-encapsulated to the point where they could form capsules and be deadly to that mouse. So these are the 4 scenarios with Griffith's experiment. I will ask you questions about this, so make sure you understand it. If my explanation was not to your satisfaction, if you needed a little more information or a little bit more clear definition, you can go back a slide, and this tells you exactly what I said, but in a more succinct manner. Okay. So transformation. Picking up genetic material that's extracellular, meaning outside the cell. Lastly is transduction. Transduction is the introduction of new genetic material by a bacteriophage. So yes, bacteria can actually get infected with viruses. We call those viruses bacteriophages. They're really small. So there are viruses that infect humans. There are also viruses that infect bacteria. We call them bacteriophages. And sometimes bacteriophages can accidentally pick up bacterial DNA and transfer it to a new cell. It can happen sometimes. How? Well, this is pretty simplistic. It's a bit more complex, but they break it down into a simple form. What they do is the bacteriophage infects the bacterial cell, and for viruses, they take over cells. That's how they get things done. Viruses don't have the mechanisms to copy themselves within their capsids, meaning the head. They don't have that ability. So they hijack the cell that they infect to do the work for them. And it's the same thing with human viruses. Human viruses don't have the ability to replicate themselves, which is why they need a live host, because they hijack the machinery necessary to copy themselves. In this case, the phage is copying itself using the host material, But on accident, one of them accidentally picks up a piece of the host DNA. So it accidentally packages some of the host DNA. Then when all of these exit this cell, when the cell ruptures and the viruses are released, this one right here that accidentally picked up the bacterial DNA will infect a new cell, and it will infect the cell by inserting bacterial DNA instead of viral DNA, which for the new cell could be beneficial because maybe the phage accidentally picked up a trait that makes it pathogenic, or it accidentally picked up a trait that makes it have the ability to metabolize a sugar it couldn't metabolize earlier. But there may be new functions, is what is important. There may be new functions. And this new cell that gets this bacterial DNA may acquire those new functions. So bacteriophages can introduce host DNA or bacterial DNA on accident. This is kind of a little bit more in depth. Basically, infected by the bacteriophage, the phage DNA recombines with the host genome or the host chromosome. Then, at some point, the phage DNA is cut out from the host chromosome, but cutting that piece out or excising the phage genes, sometimes they accidentally take some of the host genes with them. They don't just have phage genes, they also have bacterial genes. And you can see in 4 here, all of these are infected with phage. All of these phages have phage DNA and bacterial DNA. So when they're released and they infect a new cell, all that genetic material will get incorporated into the new host's genome. And now the new host has not only phage DNA inserted, but also new genes of bacterial DNA. Now, it does not mean the bacterial DNA has genes that are 100% functional and they are exactly what a new cell needs. Sometimes, if not a majority of the times, it ends up being useless. However, on the chance that it's not useless, let's say there's pathogenic genes on there, or genes that are pathogenic, the new cell has a new ability, which is a good thing. So it's important to understand that phages can transmit bacterial DNA on accident. So that's the end of this chapter. I know this chapter is relatively long, which is why it spans 3 class periods. I am going to upload this video, and then I will upload one more video with just the processes of replication, transcription, translation, and I will also do repressible operons. So I will use a whiteboard function, and I will draw that stuff out when it comes to those processes. Okay? So be on the lookout for that. This is going to be uploaded very shortly. Remember, space this out over several class periods. It's not a good idea to watch this all in one sitting, right? It's 154, so an hour and 54, so that's what? 110 minutes. So that's 2 class periods, a little bit more than 2 class periods, plus the video that I'm going to provide next. So please space it out. Okay. All right. Have a good day, and if you have questions, please come to office hours. If you can't make office hours, set up a time to meet with me, because this is difficult material, and I'm happy to answer any questions that you may have. All right. Have a good day. Enjoy the rest of your long weekend, and stay safe.